Okay, every quarter, we in Bioengineering 599, J, we try to have one session dedicated for former student, alumnus, and then the other session dedicated to current students. And today is that session dedicated to current students. So we have two PhD candidates hoping to finish their PhD this year, presenting 20-minute presentation. First speaker is Gretchen Benex. She is a PhD candidate in bioengineering, working with Viola Vogel, and she has a BA degree in Asian philosophy and religion from Dartmouth, and then BS degree in chemistry, and then hoping to get PhD degree in bio this year. And she will give us a presentation with the title of Visualizing the Fibronectin Conformational Changes via Fluorescence Energy Transfer. Gretchen.
Well, I need to put that on my slides, right? Oh, all the things are coming. Shall we hit the, do I hit the lights from up here? Okay, everybody's got it? Okay, lights out. Good, okay. Whew. Cool. So let me, I want to introduce this not by necessarily talking about fibronectin. I want you to think of this as a more general technique. We as engineers have this vast array of tools that we can use to study cells and cultures and proteins. And I want you to think of this in terms of one that fills a niche that needs to be filled, okay? So those of you who are interested in cell biology, cell signaling, it's become very clear that the extracellular matrix, the architecture of the extracellular matrix is very important to how cells react to their environments and how, they react to ligands, growth factors, etc. Those of you who are biomaterials people know that when one of the things that happens when you put a biomaterial in the body is the first thing that happens is you get a monolayer of proteins on it, and then cells come down and they interact with those proteins, okay? So you have this intermediary that is very difficult to get at in terms of what it's doing, what its conformation is. A monolayer of proteins isn't much, yet the surface affects the monolayer of proteins, and that protein affects the cells. And in the past, there's been a great deal of difficulty getting from surface chemistry all the way up to cell behavior. And this might be just one more tool we can utilize to try and get at that question. So before I go into our results, like I said, I'm trying to make this a little more general. I want to make sure that we're all on the same page in terms of our knowledge of fluorescence microscopy and the technique that we use, which is fluorescence resonance energy transfer, or FRET. So let's see if I can get this. Okay, fluorescence microscopy. I'm sure most of you are very familiar with it, but I just want to make sure that, oh, that's not very good, is it? So how it works is you generally have a fluorophore, an external fluorophore, a dye molecule, that you conjugate to a protein, either directly or through immunolabeling. And that dye molecule has an excitation here. This is essentially this is Oregon green I'm showing you, and its excitation is at a lower wavelength than its emission. And I won't give you all the mechanical, quantum mechanical information about why that is. I'm assuming you had that class. But just remember that. So standard labeling techniques can yield, what, did someone have a question? No. Yield protein location and concentration. You can also use immunolabeling with monoclonal antibodies to get some idea of conformation on surfaces through the exposure of specific epitopes. There are some disadvantages, though, because a lot of times you can see the exposure of epitopes, but you don't know what the rest of the protein's doing, okay? It has an RGD sequence, it's exposed, but what does that really mean? What's the rest of the protein doing? Because a lot of these extracellular matrix proteins that we're working with are pretty large and have a large number of different recognition sites. So in fluorescence resonance energy, or FRET, you actually use two dye molecules, okay? One is termed the donor, and the other is termed the acceptor. And what's important about this is that you choose a pair so that the absorption spectrum of the acceptor overlaps with the fluorescence emission of the donor, okay? And so what that means is here you have your Oregon green emission. Here is your, what we use is tetramethylrhodamine absorption. See how they overlap here? So if they're within a certain distance of one another, Well, you excite the donor with, in this case, a blue wavelength. That excited donor, then it's like an antenna, and the two of them start getting excited in resonance. And the donor passes that energy to the acceptor without radiating a photon, okay? And then when the acceptor reaches its ground state, it emits a photon. And that photon is in a much longer wavelength than would normally be associated with, for instance, fluorescence. It's not green, it's red. So the donor and acceptor, there were certain requirements besides just the overlap. They have to be within 10 to 100 angstroms of one another. After that, energy transfer doesn't really occur. For all you techno-dweebs out there, it goes the distance efficiency relationship goes over 1 over r to the 6th. Okay, so it's not linear. You have typically the best efficiency is about 50%, and that's for our particular diped, that's about 65 angstroms. That's where you get your strongest signals. Okay. So then you have to think, well, this is not a new technique, which I'm talking about. First described it in 1948. But with improvements in dyes and different applications, people have been using it for a while now to look at association of, protein-protein associations, trying to get distances within a molecule instead of using on proteins that you can't crystallize. That has some artifactual problems that I won't go into, but what it's really powerful for is looking at conformational changes or changes in distance in molecules. For instance, what happens to a certain region of a molecule when ligands bind. And this is just one example. So hypothetically, if you're looking at a molecule and you're saying, well, how can we look at why is it that we can look at conformational change? How do we do this with a molecule? And so say you have a protein that's all folded up. And you have the donor and the acceptor, seen here in green and red, and they're pretty close together in this particular state. And as you can see, if it's less than 100 angstroms, you have the possibility, a heightened possibility of energy transfer occurring. And so what you'll do is you excite in the blue and you see red. Now here, if somehow you could take and unfold the molecule, just unfold it end to end, you can see these are in the exact same position as they were before, but the distances, the average distances are much greater. And so the probability of energy transfer goes down considerably. And then you can even further extend this out and denature it, but it's a little hard when it's in a random coil to know exactly what the distances are. But hypothetically, you can look at increasing donor-acceptor distances as energy transfer is reduced. Okay? So the protein that we're using is fibronectin, and there's a number of reasons for that. Fibronectin, which I refer to it as FN. It's a long name. Fibronectin is very important, first of all, biologically. Okay, it's a very important component of the extracellular matrix in times of tissue remodeling, such as wound healing, embryogenesis. And it exists in two basic forms in the body. You have a globular, biologically somewhat inactive molecule that you find in blood and other bodily fluids. And here the recognition sites, biologically active sites, are masked. Okay, it's all folded up. Then cells are able to, unlike collagens and laminins, fibronectin cannot self-assemble. Okay, so cells assemble these fibrillar matrices of fibronectin, and this is where the majority of the biological activities are expressed. And you have protein extension and/or elongation. There's cryptic sites that are exposed. It may be the result of the selective denaturation of certain modules. That's also an open question. But this is where its recognition sites are unmasked. So function follows conformation, okay? And then there are some interesting questions about surface-absorbed fibronectin. For instance, on some sort of biomaterial surface or a non-biological surface. Through use of conformation-sensitive antibodies, they have been able to describe, yes, we see the similar epitopes opening up to what we see in fibrillar matrices, but is the conformation actually the same? Well, that's something that nobody's been able to answer, and we can answer that at this point, okay, at least for our particular system. So you have protein extension, the exposure of certain functional sites, but what about the variation of the function and conformation with surface chemistry? Okay, how can we get at that? And if we can get, maybe we can get a fingerprint of what it's doing on particular surfaces and correlate that with cells. That would be really interesting. We could do that. And we're working that. Before I give you the results, I just want to say that what we use is not a standard fluorimeter. We use a microscope that is attached to a spectrometer, an imaging spectrometer, that is then attached to a CCD. Okay, so we can either take images, or we can take these images and center them basically, and focus on one particular attribute of the cell, and we can take spectra. Okay, so that's the unique aspect of what we're doing here. One of the unique aspects. Okay, so the first thing you want to do is look at proof of principle. Fibronectin has some well-known behaviors under different conditions. Can we detect them? And this slide is meant to say yes, we can. So what we do is we did a spectral analysis of fibronectin in solution and absorbed to untreated glass. And we used a very low quantity of the doubly labeled fibronectin with a tenfold excess of unlabeled fibronectin in PBS. That's particularly important when we absorb it to glass. And the reason we do this is we want to make sure that there is no transfer of energy from adjacent proteins. We just want to look at energy transfer in the single molecule, intramolecular energy transfer. And we have found that this is effective in this kind of ratio. So the first thing you see is solution. In the solution, you see a very high acceptor peak, which means that there's a lot of energy transfer going on. I have normalized all this to the donor peak so you can better see the differences in the energy transfer. And then we take the same protein and we denature it, and boom, down it goes. This peak goes down dramatically. Okay, that shows that we're very sensitive to at least elongation or the conformational change in solution. We also took the same solution, it was not denatured, and we absorbed it to glass for 30 minutes. And here's very interesting. You find this intermediate state, where it appears to be unfolded but not denatured. That's what we think at this point. It's certainly not completely denatured. This here is just to show you this is a dye mixture absorbed to glass, and you have a little tiny bump here, and that's direct excitation, and that's not a whole heck of a lot. It's less than 10% of the entire. If you remember the 404s that I showed you before, the donor is going to have a tail here anyway. Okay, so that's going to come down here anyway. So there's only a little hump. So we really did not correct for that, as we're looking at the relative changes anyway, and it's very small. So now we get to do the fun part. We get to feed this stuff to cells and see what they do with it. We use NIH3T3 cells. We absorb fibronectin that is not labeled to glass surfaces so that we can reduce background absorption. Let the cells stick, and then add the serum, growth medium, and fibronectin in this tenfold excess with unlabeled fibronectin. And what do we see? Well, this would be more dramatic if the light was off, but we see some really beautiful stuff. Let me see if I can focus it a bit better. Could you? Maybe. Nope, wrong way. This one? That one? There. Okay. See? That's cool. So anyway, what you see is this, and this is what you see in the ocular. Okay? It's just gorgeous. You see these very red surfaces of the cells, and then where they're making matrix, and this is very much what matrix looks like, it's bright green. Okay? So if you take the spectra of these different components, you can see that the cell surface looks very much, and I've shown in black here, like the globular fibronectin we saw in solution. The matrix, however, although it is significantly reduced in its energy transfer, is not completely denatured. So it's unfolded to some degree and extended, okay, but it's not completely denatured. Now, if I had made this slide more confusing than it already is, you would find that the fibronectin that was absorbed from solution to just the glass surface is intermediate between these two states, okay? It's in a different, the donor acceptor distances are in between these two. So matrix proteins appear green due to low energy transfer. Cell surfaces appear red because the energy transfer is much higher. So what we assert is that the matrix and surface-bound proteins represent different fibronectin conformations, so coexisting conformations in the same cell culture. So we have red being compact and green being more extended. So what else can we learn from this? Well, we can do some cool things. We can look at matrix assembly. And this is I don't know how much you know about fibronectin, but it's quite an open question. There's a number of different competing models out there for how fibronectin is actually assembled, and they're all very complex. So just to show you what we see, here we have 1 hour. We have rounded cells, and those cells have not quite begun to make a matrix yet, but you see these interesting clusters. And these clusters of fibronectin that are in a different conformation than diffusely absorbed protein clearly, they're probably not aggregates. And the reason we know they're not aggregates is that they don't exist anywhere except for on the cell surface. It's not like labeled fibronectin falling out of solution. And we've also had that happen before, and they're bright red. So this is not the same thing. Then in about 2 hours, these aggregates have or clusters have moved to the side, and you see matrix being made, nascent fibrils kind of going centripetally around the circumference of the cell. And that's very in line with what people have found for this particular cell type. I'd refer you to a recent paper by Kenneth Yamada about fibrillar adhesions if you have any interest. By 4 hours, you have a really nice matrix being associated with the laminopodia. And in fact, even though it's very bright red here and it looks like there's no diffusely absorbed protein here, it's just that it's very low. There really is. And then by 24 hours, you have some areas of really nice confluence. Let's see if I can get that better. And you can see cells in various states of matrix assembly. And look at this beautiful matrix, just mesh web-like thing. It's great. And then you can also see more of these little clusters here, and some cells that aren't really doing anything. Okay, so this was not. These are all different samples. You know, we fixed them at different times, and we didn't try to synchronize them in the sense that we starved them in serum and didn't want them to proliferate. We just wanted to see what they were doing. And this supports a model of matrix assembly where you have globular fibronectin from solution coming down and binding to the surface, possibly integrins, although that's, again, a controversial topic. Then you have fibronectin undergoing a conformational change, which allows integrin clustering and also the opening up of fibronectin binding sites. So you can open it up, and there's on each arm of fibronectin, there's an RGD sequence which can bind a cell surface integrin. And then if you start clustering together, as they seem to be here, you get these large numbers of integrins coming together, and that's when they attach to the actin cytoskeleton. It's been shown that when fibronectin is just diffusely absorbed to one integrin, unless the integrins are clustered, you do not get that cytoskeletal attachment. After they attach, then they move to focal adhesions, and they're polymerized into extensive networks. Okay? So, what is this? What does this this technique get us? Well, first of all, it's obvious you get some really nice enhanced image contrast, but you also get this conformational mapping. So on the top, what I've shown is, let me see if I can focus that a bit. Nope, wrong way. The blue is the excitation, okay? And so what you see here are the different channels. You have separate filter cubes that are all appropriately chosen for the fluorophore that you're using. You have blue excitation, and this is the energy transfer, the red. Here's blue excitation and donor-only emission. And when you combine the two, you get an image like this. I don't have the scale bars up. Sorry, I was sick, but this is about 15 microns across. Now, if you take the same sample and you simply excite the rhodamine, this is what you get. You essentially get what you would have off of standard fluorescence microscopy, the conventional microscopy. And you do see some nice contrast, but you don't get the same kind of information. The contrast is better here, and you get this ability to map where the conformations are different. So, let me move on. So what you can do is you can almost get a fingerprint, a conformational fingerprint for a spectral fingerprint for a specific conformation, which of course depends on your protein and how you've labeled it. So in solution, we have a globular protein with strong ionic interactions holding it together. It has very high energy transfer, and when it's denatured, threat decreases dramatically. Same with cell surface absorbed. You have very high energy transfer. Fibrillar matrices the biologically active form of the protein has some of the lowest energy transfer, except for the denatured fibronectin. So we definitely have a lot of extension going on. But on surface absorbed fibronectin, even though you have the exposure of those epitopes I told you about, you have a different conformation. Okay? So that says something there already. These are not the same. And they may be giving different signals to your proteins, or to your cells. So I think this should be of general interest because it can be done with other cell types and probably with other proteins. And I think it would be really fun to do this in conjunction with, say, micropatterning certain chemical, different chemistries on the surface, letting the cells go and look at cell signaling, because we now have GFP, you can make GFP protein fusions to look at signaling. So it has a lot of applications in both cell biology and for biomaterials people. So with that, I want to thank all those people who contributed this, particularly Lauren Ba and Li Chia Cheng. They were great. Lauren and Li Chia worked on the optimization of the labeling and the cell culture. Of course, Viola, always there, so I can go scream to her, I don't understand. And the Vogel group for the same reason. And then it's always nice to have funding. For part of this, I was on an NSF fellowship. NIH also funds this, and the cardiovascular training grant now funds me. So thank you.
Any questions?
Can you interpret the results on the basis of where the dyes are in the molecule?
Yes and no. We have tried various strategies for labeling, actually 3. One is random on just lysines. The other is 50-50 on fibronectin has 4 cysteines that can be specifically labeled after denaturing it, okay? And we've done this with a 50-50 mixture. That's not what these are, but we have gotten information off of that, and yes, we can make some statement about that. To get the best conformational information, we don't know where the lysines are yet. But we're also, so we can say that the conformation is changing, the distances are increasing, but we cannot tell you exactly by how much. Whereas in the ones that we have just the SH or the cysteines labeled, we can tell you that the fibronectin, I didn't have time to talk about it, but it is extended so that there is really no energy transfer between them. Okay, so we know it's in a very extended state, more extended than on the cell surface, on just the glass surface. We're working also with in a collaboration to do mutagenesis so we can specifically label different regions of the molecule.
Before I ask my second question, I want to make sure it makes sense by asking, between the upper surface of the cell, intracellularly, and below the cell, above the substrate, where do you believe the majority of the fibronectin is concentrated for all 3? Okay.
The diffusely absorbed fibronectin is concentrated on the membrane surface on the top, the red stuff. Okay? That's where we have to focus to get the appropriate spectrum. However, the majority of all of what you saw in terms of the green is on the bottom surface. Although, if you have many cells, you do get fibronectin matrix formation on the top of the cells, but you can distinguish. So you can actually change the focus and see where the red stuff is, where those little spots are that I showed you, those little clusters on the top, and then the matrix is below. It's on the bottom. The majority of the matrix, I should say, on the glass surface.
Do you think that the concentration is higher under? This is getting too long a question.
Okay, I'm sorry. We can talk about it later. I'd be happy to. Okay.
Thank you, Rachel.
Thanks, everybody.
Next speaker is Todd McDougall. About 5 bioengineers tried to work it out to have a good connection with a video projector and failed. So he will be using a kind of new model presentation using the camera up there. So you will not see anything here. You have to see the monitor. Okay. Todd is a PhD candidate in bioengineering. Again, hoping to finish his degree in fall, but I heard 6 months ago wanting to finish it in summer. So now it's...
No pressure.
And Todd got his the BS in engineering degree in biomedical engineering and electrical engineering from Duke in 1997. And then he came to UW Bioengineering. So he'll be finishing his PhD in 4+ years. So remember, it is possible to get PhD in 4+ years. And he has a number of honors and awards, like first place 2000 Rushmore Lecture poster competition last year, and honorable mention in 1999 ABS student poster competition, and number of other awards. And he also served on the departmental student advisory board and provide us a lot of good input to the department and also liaise with the students. So today he will give us a presentation with the title of the Spatially Organizing of Cardiomyocytes for Myocardial Repair. Start.
So thank you very much. I apologize. The laptop presentation didn't go quite as planned, but we're still going to go with it from here. So I apologize. The resolution of some of these isn't going to come up quite as nicely as I had hoped. So, as many of you are probably aware, cardiovascular disease, and specifically cardiac heart failure, still remains one of the leading causes of death in developed nations. In the United States alone, it accounts for more than 1 million new heart attacks and greater than 500,000 deaths. And the main problem with this is that once the myocardial tissue, so the tissue of the heart, is damaged, this is a permanent wound that gets, or a permanent scar is formed. And this is because the heart itself doesn't have any capacity for regenerative processes. So certain techniques have been tried to try and fix the damaged heart, including heart transplantation, so simply try and remove the pre-existing heart and implant a new one. However, one of the main problems with this is simply the availability. There's only about 2,000 organs that become available for transplant annually, and this clearly is far too few for the number that are needed. And another, which is more of a bioengineering attempt, is to try and artificially create a heart. But this also suffers from a number of complications, which, at least in the short term, are going to take a tremendous amount of time, and a number of hurdles have to be overcome before they would actually become feasible. So these are actually used, though, but most of the time only for a temporary, as a bridge to transplant until someone can actually obtain a heart. In recent years, however, new thought has been applied to this, and there have been some new insights into how could we possibly try and regenerate some of that which is lost. And one of these approaches is referred to as cellular cardiomyoplasty. And by this, I simply mean the injection of cells directly into the heart, targeted towards the wounded area, that could then hopefully be used to try and restore some lack of function. And as one step kind of beyond that would be to actually try and engineer a piece of this type of tissue that you could perhaps graft into a heart. And that's what I'm going to talk about today, is my work and how it's oriented towards cardiac tissue engineering. So as you look at the heart, it's clearly a very complex organ, and to be able to make a structure like this, it's important to be able to first study that structure. So it's important to be able to identify what types of cells you would need, what types of material you would have to build this tissue upon, and then once you have those materials, how will you assemble them together properly into something that looks like the actual heart or piece of the heart. And thirdly, or lastly, how will this actually perform, both aside from the tissue, so in vitro, when you're actually making such a tissue construct, but also once it's actually grafted into a host tissue, how can you ensure that it would respond properly? For today's talk, I'm really just going to focus on assembly. So how can we take cells, and what are some methods we could use to organize them in such a way that you might be able to recapitulate some of the structural features that you see in the actual myocardial tissue. So as I said, this is the heart, as you start to study, it's really quite fascinating, and it's apparent very readily that this is a highly organized structure, and that this structure is is inherently related to the function of the tissue. And so if you were to take a look at the section of the heart, try and use this, such as here, and you were to go from the inside of the heart to the outer wall of the heart, there's a number of individual sheets of cell, sheets of the cardiac muscle cells that have a precise type of orientation both within those sheets, and then those sheets have a particular arrangement with respect to one another. If you were to take a look at these cells here, and again I apologize because you're not going to be able to see with the resolution, but these are the individual cells. You can see that they're primarily oriented in a single direction within one layer of the tissue, and that if you, under higher examination, you can actually see the myofibril, so the structures that are actually responsible for force production within these cells, you can actually see that these are also aligned with this same, or with the same direction that the cells are oriented. And there are also these sites, specialized sites of cell-cell junctions that you find typically at the bipolar ends of adjacent cells. So this clearly has a very distinct structure, and as I said, this is important towards the actual function of the heart from this individual cellular basis as it assembles itself into a larger tissue and then the complete heart. So how can we achieve or hope to achieve the same type of spatial control that biology has so elegantly demonstrated? And by this I mean how can we provide the types of spatial instructions or cues to cells to induce this organization, both intracellular organization, but also as you take individual cells and start to add them together and assemble them into a tissue and then larger organs. Developmental biology can give us some clues as to how they do this, how this is done properly, because it's developed a very sophisticated way of using spatial temporal patterning of molecules. And this allows the cells to be able to take the cues from their environment and thus be able to organize into tissues, and as you can even see here, into complete organisms that are very highly organized or compartmentalized. One technique or one method which I'll be talking about today in which we can try and replicate some of these features is using micropatterning. And the advantages of this technique is that you can actually work at the size scale on the individual cells, or even as Gretchen was just alluding to, you can work at even smaller levels, nano levels perhaps, to be able to manipulate the molecules that are responsible for this type of organization. One of the disadvantages largely at this point is that this is micropatterning techniques are largely applied in 2 dimensions, whereas you can clearly see that this type of organization actually in vivo or within an organism occurs in 3 dimensions. But as time proceeds, I think we'll see more and more. There are some more preliminary attempts to be able to engineer in 3 dimensions, and I think that this will be the expansion of that will allow us to develop even more sophisticated techniques. So today what I'd just like to focus on is the method that we've been using to do micropatterning and how we're applying it towards cardiac tissue engineering. The method we use is referred to as microcontact printing, and by this technique we begin with a standard photolithographic patterning method whereby we create a template on a silicon wafer that has micron-sized photoresist features. We then can cast a polymeric stamp against the surface of this template, and the stamp will then the stamp features will then be the exact mirror image of the features we have on the wafer. We can then remove this stamp, and in the second step of this process, we can actually use a protein solution, adhesive matrix protein solution, and for the images I'll be discussing today, I'll be using laminin for all of these experiments to ink our stamp. So we coat the surface of the stamp with the protein, we rinse it and dry it, and then bring it into contact with the substrate that we're working with. And what happens is you're left with this resulting protein pattern where protein is deposited just in the regions of contact between the stamp and your surface. So it's a relatively simple technique, and we've actually been able to use this for not just laminin, but a number of other adhesive matrix proteins as well. So these are the types of patterns that you can achieve with this method. The nomenclature I have here is either 15 by 20 or 30 by 20. The first number refers to the lane width, so these are 15 micron wide patterns. And the second number is the spacing, so this is the space between. And we can use a different series of these to either make these lanes wider or more narrow, and also the spacing between. So we can engineer that component of it quite easily by our microfabrication. And which we have here are patterns of fluorescently labeled protein that the protein's been fluorescently labeled so we can easily visualize it. We then seed cells onto these. In this case, we're working with cardiac muscle cells. These are rat from primary isolation of rat neonates. And what you can see is that the cells are highly aligned by the pattern, and that their adhesion is strictly regulated pretty much to the regions of the pattern. I say pretty much because there are some regions actually where you can see that there's some bridging between adjacent lanes. However, this is some type of factor that we can actually control by changing some of our spatial patterning dimensions. So we've also analyzed these to take a look at how cell shape, how we can engineer or change the cell shape with regard to simply changing the patterning width. And when we're working within a range of about 5 to 30 microns, so this is on the order of individual cells, and specifically in the order of the width dimensions you would typically see of adult cardiomyocytes, you can actually find that the corresponding aspect ratio measurements that you can take by measuring the long axis of the cells as well match pretty well with the types of morphometric analysis that has been achieved by looking at cardiomyocytes in actual tissue or by isolating myocytes from actual tissue. So what this suggests is that we actually have ways to be able to engineer these cells to have the same types of dimensions that you would actually see in tissue. If we start to look at within the guts of these cells and how they're organizing, one of the things that's really apparent is we can see the differences in the organization of the myofibrils. And what you can see in the far left panel is that with unpatterned cells, so these are just on coated dishes as are commonly used, you can see that the myofibrils, which are these filamentous structures here with the bands, these dark bands are the Z bands, try and give you some orientation. They're largely in disarray. There's really no clear order or organization to these. Whereas in contrast, you can see that in the pattern cells, that these are actually highly aligned. In this case, the pattern lines were going up and down here with the same direction of the myofibrils. And that the Z-bands are often in registry over rather large distances, which you can also see is comparable to the types of properties you would actually see in the neonatal heart and also in the adult heart. So this suggests that by organizing these cells, we're pushing them towards the type of phenotype that looks more like actual adult heart tissue. So one of the other unique characteristics of cardiomyocytes is that they have specialized cell-cell junctions, which I refer to, and these are intercalated discs. And at these are very important because this is the site at which the electrical mechanical coupling of adjacent cells occurs. And in this here, you have several different components that are localized at these sites. And first of all, cell-cell adhesion molecules such as N-cadherin. You also have cells electrically coupled by gap junctions such as connexin 43 in the ventricular myocardium. So we were interested in, first of all, since we saw that the shapes of these cells and some of their morphometry look similar, to also analyze the expression of these molecules and how they were and where they were expressed in our pattern cultures. So again, if we're comparing the pattern cells to the unpatterned cells, and in the top row we're looking for N-cadherin expression, which appears in a sort of greenish-yellow color, and in the bottom for connexin 43. And what you can see is that in the unpatterned cells, you see the expression kind of circumferentially distributed around the edges of adjacent cells. Whereas in comparison with the patterned cells, you can see that we have these localized bands of expression primarily oriented at the ends of adjacent cells because we're controlling how they contact their neighboring cells by the patterns. And that you see a similar type of expression pattern for connexin 43, and that our pattern cultures closely resemble the types of expression that I was referring to for sections from an actual adult heart. So again, these types of features suggest that these cells are morphologically resemble actual adult tissue. So one of the fun things about working with these cells is that they actually beat in culture. And this probably isn't going to show up very well with this is probably the most commonly used material right now in tissue engineering. However, there's a number of disadvantages possibly to its use. One of them, especially in this case, or for soft tissue applications, is that it doesn't necessarily possess the right mechanical properties. It's a rather brittle, rigid material that might be more suited towards hard tissue applications. So recently we were excited to find that a new class of polymers, biodegradable polymers, has been developed by Dr. Kim Woodhouse at the University of Toronto. And these polymers are unique because of the fact that they are biodegradable, but they also have a rather elastomeric in nature. So this might be the type of material that might be more suited towards the soft tissue application, especially something like contractile muscle tissue. So we've begun collaborating with Dr. Woodhouse and using some of these materials in our research. And so what I have here is simply a picture. These are small 10 milliliter Teflon beakers. And what we do to work with this polymer is we dissolve it in an organic solvent such as THF, and then we cast it in the beaker, and then we can remove it once the solvent evaporates. And you're left with this small, clear, pretty flexible film that's relatively thin, and that can be controlled basically by how much polymer or solvent you're adding when you're casting these samples. And then what we do is we pattern them much in the same way that we print onto our polystyrene dishes originally. See these little discs as they kind of appear with a high density of cardiomyocytes, and then we can continue to culture these for several weeks, and we've kind of studied them over that kind of time course. So I'd just like to show some of the interesting results we found with that. What I have here is these are two images of a sample that was fixed after about 4 days, and what we have on the left is the cells, you can tell, are highly aligned. They're stained with phalloidin, which stains the actin filamental. So this will stain all of the cells that are in this culture. And I say all of the cells because although the isolation begins with primarily greater than 90 to 95% cardiomyocytes, if you're left untreated, since there are contaminating fibroblasts in there that will overgrow your culture. So to control that, we can actually use a small drug that inhibits proliferation. Since the cardiomyocytes are non-proliferative at this stage, we can selectively inhibit the fibroblasts and then study the effects on the cardiomyocytes. And so on the right, what we have is a stain with an antibody that's specific for muscle cells. And so what you can see by comparing these two is that what you're left with is a highly aligned, very dense culture of cardiomyocytes. In this case, using line patterns about 15 microns wide, and again, only about 10 microns apart. So you've packed a lot of lines in and oriented the cells, but packed in a lot of adhesive area for the cells as well. And one of the interesting things with this is that we've, there it is, then also looked at, you now have an elastomeric substrate, and how will cells, contractile cells, behave differently on an elastomeric substrate compared to the type of rigid substrates that we've studied in the past. So to orient you, I have a note on the bottom that you're going to see some rings structures in this, and this is a result of actually from being cast against the Teflon beakers. There's a similar type of ring, as you can see here, that we pick up from the morphology at the bottom of the beaker. But at even low magnification, you can tell that these cells are actually able to contract this material. And as you zoom in a bit on each of these, what you can see is that these cells are aligned in this direction with the initial pattern lines. The phase contrast is distorted by those rings, so it's hard to see the individual cells. Plus, it's kind of it doesn't remain a very flat substrate, so it's difficult to get one plane of focus as you're visualizing. But if you were to compare the type of contraction you see here with what you saw previously, that's actually a similar field of view. So you can tell that the magnitude of this contraction looks significantly greater than what you would see on a rigid type of support or material underneath these cells. So one of the other things we're interested with this is how can we actually then take something like this and actually apply it towards tissue engineering. And so to do this, we've begun to examine the feasibility of using this type of material with pattern cardiomyocytes to actually graft onto the heart. And in this case, we're using I highlighted here the outline of this piece of the material. And what we've done is we've applied just a thin fibrin gel that kind of helps to seal it onto the surface of the heart. And this is onto the left ventricle of a nude mouse. So some of the questions that we're interested in answering by this is, first of all, what is the type of host response you would see from this material? Can this be used as a suitable delivery vehicle for myocyte transplantation? And also, how well do the cells remain aligned, since that's really the crux of what I've been studying is, and will that even matter once you're in vivo? And just a simple example, this is one animal that was sacrificed after about a week, and this is just the histology. I can see, well, this is actually from the left ventricle. There's a large immune response, or foreign body response, sorry, since this is a nude mouse, foreign body response along the surface of the heart in response to just the material alone. This was with no cells, kind of as you would expect. But you can also see just a remnant here, actually, of what was left of the polymer film after processing. And there's a thin collagenous kind of capsule starting to form around the edge of this, again, as you would kind of expect. So this is just some of our preliminary pilot studies to see, can we go farther with this technology. So some of the conclusions of this is, first of all, that we believe that these micropattern cultures could be suited to a number of different applications, simply because that the organization of the cells appears to allow us to recapitulate the type of morphological features you see in the native heart. Also, that it's quite clear that the aligned cardiomyocytes can actually contract on a more suitable substrate like the biodegradable elastomeric polymer compared to some of the more rigid ones. We've also done comparisons with PLGA. I didn't have time to show you that today, which you wouldn't see that type of contractional activity. And also that these patterned polyurethane films may serve as a good carrier towards or using for myocyte transplantation studies. So with that, I would just like to thank my advisors in their labs, a number of people who have, from those groups who have worked with me on this project, as well as several of our collaborators on this project and other projects that have been related to this. So thank you very much for your time.